Next speaker is Harry Melnick from Weill Cornell Medical Center in New York. He's going to tell us about BCL6, another master transcriptional regulator.
Well, it's really a pleasure to be here. I want to thank Hugues for the invitation to this meeting on my favorite topic of transcription factors in cancer. And for this talk, I'm going to focus on a transcription factor that is called BCL6, which is a master regulator of the humoral immune response. And I just want to start by reminding you that it is somewhat of a paradox that we are able to survive in a world where we share our environment with a whole series of organisms that evolve very rapidly, much faster than we can. And yet we can fight them off when we are infected by them. And so there's no way that we can actually have any kind of defense against this diversity of organisms, and which evolved during our lifespans. And so we've had to come up through natural selection with a mechanism that can match that rate of evolutionary speed. And the response to this challenge has been the development of a pathway for B cell development, whereby mature B cells, when activated, can essentially put on pause the classical B cell transcriptional program, migrate to the interior of lymphoid follicles, and replicate at the fastest rate that's possible for human cells to divide, while at the same time undergoing mutagenesis of their immunoglobulin loci. This clonal expansion and mutagenesis leads to the stochastic development of clones of cells that encode for an antibody that has acquired affinity for a new invading organism. And those cells can then be selected by T cells to undergo terminal differentiation if they've been successful in creating a new high-affinity antibody. And those final plasma cells that come out of this will be the ones that produce the antibodies that can clear these new antigens. But of course, the vast majority of these cells fail to do this and undergo cell death. And the master regulatory factor, the transcription factor that makes it possible for this setup to occur, is BCL6, which gets turned on as these early germinal center B cells migrate to lymphoid follicles, and then gets turned off again upon receiving the T cell signals that will select B cells for terminal differentiation. And from the cancer standpoint, we are interested in this because a great majority of immune neoplasms of lymphomas arise from these particular cells, which you can easily, I think, understand because of their mutagenic proliferative state. They're kind of like a cauldron for cells to acquire mutations off target in genes that would lead to them becoming transformed. So, and these tumors uniformly express BCL6, and in fact, BCL6 is the gene that's required. It's their fundamental lineage factor for all these different tumors to maintain their survival. So this factor was first cloned as being frequently translocated in diffuse large B-cell lymphomas, which are the most common type of lymphoma, and they arise from germinal center B cells. It encodes a BTB zinc finger transcription repressor. The knockout mouse shows that it's required for forming germinal centers. When it's expressed constitutively, it causes the development of diffuse large B-cell lymphomas in mice, and it's expressed in the vast majority of patients that have these tumors. And so now we're going to go over some of the mechanistic aspects of BCL6 that we've been uncovering piece by piece over the years. And first, just in general sense, give you a sense of what it actually does in lymphomas. So we and others have mapped extensively BCL6 target genes by ChIP-seq and other methods. And here are some of the key functionalities that to keep in mind. One of the main functions of BCL6 is to turn off this DNA damage checkpoint pathway that includes ATR, CHEK1, p53, et cetera. This obviously makes perfect sense if you're thinking that cells want to be proliferating and tolerating genomic instability simultaneously. So it does a very dangerous thing in these cells, but that's what makes it possible for that phenotype to manifest. At the same time that it's doing this, it's also blocking the genes that are required for plasma cell differentiation, or the genes that are required to respond to T cell signals that would enable exit from the germinal center reaction. So it blocks the differentiation, and it maintains them in this unstable phenotype. And on top of that, it also suppresses immune signaling pathways such as MHC class II, CD2A, CD74, PD-L1, and interferon gamma pathway, including MHC class I, which essentially makes these germinal center B cells kind of invisible to the immune system, which is important for them to have enough time to replicate and divide and create these clones. So it's kind of like a cloaking device that hides the cells from the immune system. Normally, B cells are very immune interactive, but these germinal center cells kind of go under the radar a bit with this stealth technology, and at the same time evade checkpoints and block differentiation. So I think it's immediately clear to you why this is a dangerous thing. And in fact, BCL6 walks a very fine line, a dangerous line in germinal center B cells, where it represses all these potential tumor suppressor-like mechanisms, but at the same time, to make up for these sins, it also represses a number of key oncogenes. And so it silences MYC, it silences BCL2, cyclin D1, BMI1. All these genes are also silenced, and so it enables these germinal center B cells to be at this very precarious balance between transformed and untransformed. The reason why lymphomas characteristically develop translocations of MYC or BCL2 is to escape BCL6, is to provide those genes with a new regulatory elements that are not repressed by BCL6, and that combination of those translocations essentially just tips the balance, and now BCL6 is going to be suppressing all these tumor suppressor genes, and MYC and BCL2 are no longer under its control, as now we have a very bad situation, and that's what leads to lymphomagenesis. So now we'll talk a bit about the mechanism of action from the biochemical standpoint. BCL6 is really a virtual Swiss Army knife with many different modules that have different functionalities, and it gets quite arcane as you go deeper and deeper. So at the most superficial level, it has three different regions, a BTB pos domain at the N-terminus that interacts with three co-repressors called SMART, N-core, and B-core, a middle unstructured domain that interacts with essentially the co-rest factors and also MTA3 and NERD, and then zinc fingers that bind to its cognate DNA binding sequence and also can interact with other factors such as class 2 HDACs and ETO, although functional significance of this is not yet elucidated. To understand the relative contribution of these different domains to the phenotype of BCL6 in mouse development, we generated some knock-in animals where we point mutated the sites that are required for interaction with these co-repressors in the BTB pos domain and RD2, and compared and contrasted the phenotype of the resulting animals to the BCL6 knockout mouse that was reported a long time ago by De La Favre, Stout, and Fukuda. And the BCL6 knockout mouse has a very dramatic phenotype. It's embryonic lethal at a late fetal stage. Many of the mice die. Some are born. The mice that are born are runted and wind up dying within 4 to 6 weeks. They don't have germinal centers. They don't have T follicular helper cells. Their macrophages are hyperactive, and they manifest massive inflammatory reactions in their hearts and lungs and everywhere else, and that's what kills them. So there's loss of control of macrophage inflammation that's lethal. Now, in striking contrast, if you just make the point mutations in the BTB domain that disrupt the three co-repressors that interact with it, you get none of those phenotypes except for absence of germinal center B cells. And if you mutate the second repression domain, you also have lack of germinal center B cells, and in this case, also the TFH cells are impaired. And so there is, to some degree, some lineage specificity to the different domains of BCL6. Of course, you may be asking, how about this part over here? It turns out that this macrophage functionality is actually a passive repression effect of BCL6 zinc fingers competing with STATs because their binding site of BCL6 overlaps with that of STATs, the same sequence. And so in inflammatory syndromes or in macrophage equilibrium, the inflammatory chemokines are kept in a repressed state by BCL6. When BCL6 is stripped off after inflammatory signaling, then STATs can bind massively and mediate inflammation. Without BCL6, you lose that rheostat, and that leads to constitutive macrophage hyperactivation. To be more precise about what each of these domains does, what we know about this middle repression domain is that through co-rest HDAC2 recruitment, it represses genes S1PR1 and GPR183 that are required for B cells to be able to traffic to the interior of lymphoid follicles. And so B cells, when they're activated, are outside of follicles. Repression of these genes is required for migration in, and that is controlled by this particular domain. Once they get into the follicles, their proliferation and survival, the checkpoint repression, is mediated through the BTB domain that represses genes like ATR, CHEK1, P53, P21, et cetera, through this other mechanism. And so the mechanisms hand off to each other. But the one that's required for this expansion is the BTB domain, where we're going to spend most of the time discussing from here on. So the BTB domain shown here is the crystal structure as generated by Gil Privé, and who's a structural biologist in Canada. It forms an obligate dimer, one monomer in blue, one in white, and the peptides from SMART, NCOR, and BCOR that bind to the BCL6 require dimerization and bind here to the interface between the monomers and form this very long surface where it dives deep into the interior of the molecule with multiple intermolecular contacts. What's interesting is that although there are many BTB zinc finger transcription factors, the residues that interact with NCOR, SMART, and BCOR are only in BCL6 and not conserved in the other family members, and hence this peptide from co-repressors that binds only can bind to BCL6 as far as we can tell, having performed biochemical studies in about 20 different BTB factors and not seeing any binding to those. Now, if you go into primary germinal center B cells from humans or mice and perform ChIP-seq for BCL6 and all of its cofactors, you observe several things. First of all, BCL6 is dominantly binding more to enhancers than it is to promoters, so this white and blue is enhancers. But if you look at BCOR BCL6 complexes, those are more localized to promoters, whereas these SMART and NCOR complexes, the BCL6, are really very dominantly occurring at enhancers. And you probably know of SMART and NCOR from the nuclear hormone receptor field, where they're well-known co-repressors. They were cloned as co-repressors for nuclear hormone receptors. But it turns out that in lymphomas or in germinal center B cells, BCL6 greedily takes up all the NCOR and SMART, doesn't leave any behind for the nuclear hormone receptors, and owns it. And that may be because the BCL6 binding site for these factors is actually much, much broader than the small one that nuclear hormone receptors have. And so all of it goes to BCL6, whereas BCOR is about 50% colocalized with BCL6. And you can see there's some areas where you have all three of these components together. I should mention to you that NCOR and SMART are very highly homologous proteins that have the function of allosterically activating HDAC3 as well as other things. And BCOR is totally unrelated to NCOR and SMART and forms a typical polycomb complex. So looking more precisely at the localization of the complexes, what we observe, first of all, is that by ChIP-seq, that at gene promoters, the dominant model is BCL6 BCOR binding. Sometimes there's also SMART as well, the so-called ternary complexes. And in fact, we've shown biochemically that you can get these ternary complexes, and when that happens, that's the most repressive activity of BCL6. But BCOR is a dominant promoter complex, whereas SMART and NCOR are the ones that are located at enhancers. And so the mechanism of action at promoters and enhancers is quite different. And in fact, what's curious is that the genes where BCL6 binds to promoters are actually not the same ones where it binds to enhancers. So it seems to have two independent programs where it's a promoter repressor or an enhancer repressor, and there's very little overlap between the two, which gives it considerable functional flexibility, and that's important in the germinal center reaction, as I hope I'll be able to get to say later on. Now, another thing we noted by performing a lot of ChIP-seq studies in primary germinal center cells is that these BCL6 promoter complexes BCL6 BCOR promoter complexes tend to overlap very strongly with sites, with promoters that acquire bivalent chromatin in the germinal center reaction. That means that they acquire, these are genes that are activated in B cells, but become repressed in germinal center B cells, and by acquiring H3K27 trimethylation at the same sites. And it turns out that that functionality, making chromatin bivalent, is caused by EZH2, a polycomb protein, part of the PRC2 complex that is upregulated in the germinal center reaction. And if you knock down EZH2 or BCL6, this promoter program goes up. And so from the BCL6-centric view, it's a bit disappointing to see that it's not sufficient to repress its targets, but it actually needs EZH2 there as well. And this prompted us to and so we have EZH2 methylating histones, BCL6 recruiting BCOR. We wanted to understand if this was a related process because BCL6 and EZH2 do not interact, nor does BCOR and EZH2. We wound up finding, this is a summary of like 4 years of biochemical experiments, is that EZH2 in germinal center B cells deposit the H3K27 mark. Of course, the mark itself does not repress transcription. It has to be bound by other things. The classical EZH2 partner complex PRC1 is actually not present in these cells. So the canonical functionality for how this works is not applicable to the germinal center reaction, which is puzzling. But we did discover that, indeed, there's a BCOR forms an alternative to classical polycomb in these cells. And the K27 mark that EZH2 leaves is bound by a protein called CBX8, which in turn binds to ring 1B, which mediates histone ubiquitination, which in turn binds to PCGF1, and PCGF1 binds to BCOR, and BCOR binds to BCL6. And all this, set of interactions, you'd think, this is, why is it so complex, they're all quite weak protein interactions. Stoichiometrically, their interactivity does not favor formation of these complexes, and you require both the presence of the EZH2-induced mark and a binding site by BCL6 for this complex to assemble stably. If you don't have both sites, there is no assembly of polycomb repressive complexes. And what this complex actually does is to restrain polymerase and cause pausing at genes that's targeted. EZH2, of course, doesn't have a targeting mechanism to go to particular sites, and so it can spread, and it can essentially travels chromatin. It only forms repressive complexes where it has the sequence specificity conferred by BCL6 to anchor these polycomb complexes. And this explains, in reality, why EZH2 in germinal center B cells carries out entirely different functions than what it does in stem cells. It's nothing like stem cells. It has its own new functions that's conferred by BCL6, telling it where it is competent to form a polycomb repressive complex. So that's how the promoter aspect of the BCL6 B2B domain functionality works as a form of combinatorial tethering with EZH2 through CBX8. Now let's switch to the enhancer side of things, where there's another also very interesting aspect that was discovered by Katerina Hatzi, who was a former student. And she found that in the dark zone, in the germinal center reaction, where BCL6 gets turned on and recruits SMART and HDAC3 complexes to its target enhancers, it essentially converts enhancers that are active in resting B cells that have H3K27 acetyl to a poised configuration. And so she found that HDAC3 actually is a deacetylase for H3K27 acetyl and pauses these enhancers. They remain marked, but they are not active without that mark. And upon receiving signals from T cells, what happens is that there is CD40 through ERK winds up phosphorylating SMART, which goes to the cytoplasm. HDAC3 no longer can be recruited to these sites, and now P and P300 can reacetylate them and reestablish the enhancers that are required to respond to the signals that T cells send to B cells through the B cell receptor or through CD40 or through certain cytokines. So there's a toggling on and off, like a light switch on and off, that happens at enhancers through this mechanism and at promoters through that EZH2 mechanism we just talked about a minute ago. Now, an interesting nuance of this enhancer toggling mechanism is that it actually gets broken very frequently. So this switch is actually destroyed in lymphomas, which about 40% of them manifest somatic mutations in P or P300 that result in a reduction in the dosage of histone acetyl transferase. What this does is actually break the switch and enables BCL6, and especially downstream of it, HDAC3, to act now unopposed to maintain enhancers in this deacetylated configuration. And as a consequence, these B cells can no longer respond to T cell signals. They don't see them because their enhancers cannot react, and they become transformed. And these hits are initial hits for lymphomagenesis that occur early in disease and in tumors are manifested throughout the malignant clone. Now, if this is the case, you might predict that tumors with these mutations would be driven by this mechanism, by this HDAC3 mechanism. And if that's the case, then those tumors would be quite sensitive and amenable to being targeted by HDAC3 specific inhibitors. And indeed, just to show you a little bit of data that's unpublished, Patricia Mondello, who's a fellow in the laboratory, has found that P mutant cell lines are much more sensitive to HDAC3 specific inhibitors as compared to wild type. And here, generating CRISPR cell lines where CRISPR mutation or deletion of P, again, you make them hypersensitive to HDAC3 specific inhibitors. And there's no further benefit from using pan-HDAC inhibitors. There's no added killing, but the pan-HDAC inhibitors, of course, are non-specific and kill everything and are too toxic to give to the full concentrations in people. And one of the interesting aspects, if you're going back now to the question of B cells hiding from T cells in the germinal center reaction, well, you know, I mentioned to you that this is done by BCL6. BCL6 binds to the major enhancers for the entire MHC class II loci. And here it recruits the SMART co-repressor and deacetylates, comparing K27 acetyl in germinal centers or naive B cells, it deacetylates the enhancers, and that's what causes MHC class II to go down. And what was observed by Michael Green several years ago is that patients with these P mutations have extremely low MHC class II expression and lack of infiltration of the lymphomas with CD4 and CD8 cells. And those patient samples couldn't actually induce their T cells to activate ex vivo. Now, it was also shown by others that loss of class II is a bad thing in DLBCL, and in fact, P mutation is a poor prognostic factor. And what we showed is that P loss of function, if you just engineer this in mice, you can de novo acquire suppression of MHC class II through this overactivity of HDAC3, and you can rescue it by giving HDAC3 inhibitors. Now, as you're probably aware, the idea of giving CAR T cells is to enable T cells to see tumor cells that are hiding from the immune system. Now, the question is now, what if we could actually achieve the same effect if we understood the mechanism by which the cells are hiding? We could have them re-express those interaction proteins and have the same effect as CAR T cells by simply using small molecules to reactivate the immune signaling pathways. And so that's the hypothesis. Here's just an example of RNA-seq treating lymphoma cells with HDAC3 inhibitors. And now here we put a primary human lymphoma specimen into mice and treated the mice with HDAC3 specific inhibitor. And here we're staining for MHC class II, and you can readily see massive upregulation of MHC class II on the surface of these primary tumor cells, which you'd say, okay, would that make them sensitive now to T cells being able to kill them? So to ask that question, Patricia did the following experiment where she implanted primary human lymphomas into mice and then infused them with human T cells. The T cells had 2 weeks to get into the mouse and meet and greet the lymphoma. After 2 weeks, they start to get GVH, so you have to sacrifice them. Then Patricia purified out the tumor-infiltrating lymphocytes that had now gone into this tumor, and then separately took the same lymphoma cells and treated them ex vivo with HDAC3 inhibitor or vehicle, washed it out, and then had them interact to see if the T cells now could recognize and kill the B cells. And in fact, that's what she observed. Here you can see in the red curve T cell dependent killing of the lymphomas in the HDAC3 inhibitor exposed cells, but not in the vehicle. And at the same time, T cell activation, showing the T cells are also are getting activated. And she can actually block this by incubating the B cells with MHC class II blocking antibodies. So class II is playing a very central role in this recognition. And so we can maybe epigenetically overcome immune perturbations that we up to now are trying to do very elegant engineering of T cells to get around, or perhaps combine the two approaches together. And that kind of segues to the question of targeting BCL6. Can we actually harness this key transcription factor as a therapeutic target? And of course, one way to think about this is, yes, we can. By using HDAC3 specific inhibitors, we can hit this enhancer toggling program. And using EZH2 inhibitors, we can hit the repression of bivalent promoters. So we can, to some extent, target it just by working with enzymes that mediate its repressive effects. But for many years, we've also thought about trying to do both things at the same time, more specifically, by hitting this groove that I showed you earlier, the crystallography of, with peptides or small molecules that could disrupt both B-core and SMART binding. And over the years, we've done this first by generating peptides, which was the first example of a rationally designed transcription factor inhibitor. These peptides were very unstable and not suitable as drugs, but then we developed peptidomimetic inhibitors that could bind to the lateral groove and block co-repressor recruitment, and subsequently small molecules that do the same thing as well. The most recent tool compound that we have is a compound called FX1 that that binds right to that key pocket to the sweet spot in the surface of BCL6. And even giving this compound at low doses, this is just 25 milligrams per kilo or 50 per kilo, essentially can eradicate and profoundly suppress lymphomas in xenograft systems, underlining how critical BCL6 is as the kind of setup, the lineage factor that is required for the whole germinal center phenotype that is manifested in these lymphomas. So nowadays there are several companies that are also working on inhibitors that bind with better affinity than ours, and we also have some efforts in trying to make improved compounds. But at some point these compounds will become available, and I think they'll be interesting to test in people. We know already that EZH2 inhibitors are quite effective in patients with lymphoma, with a 92% response rate in follicular lymphomas that have EZH2 mutations, and also 30% in DLBCL, even those that are refractory. So these approaches will eventually get there and be useful in patients. Now, the last few minutes I want to talk to you about some thoughts that have arisen from studying BCL6 over the years, which is how did it actually evolve in nature? Why is it there? Why is it in us? And what does this actually mean for cancer therapeutics? So one of the characteristics of germinal center B cells is that they are tolerating massive stress as they replicate at this speed. They have oxidative stress and replicative stress and genomic stress and all this bad stuff going on. They become extremely dependent on HSP90, not surprisingly, to maintain protein folding and protect the function of the proteins that maintain the survival of these cells in these conditions. And in fact, one thing we observed several years ago is that HSP90 is present in the BCL6 complex. It's actually localized with BCL6 to its target genes with all the co-repressors, and it's essential for maintaining integrity of these complexes. We don't know if it's maintaining stability of complexes or maintaining the proteins or blocking them from becoming degraded, whatever it is, but it's required to be there. You can show it's there by chip, and if you inhibit using a drug that's specific for the activated form of HSP90, we observe that these target genes of BCL6 will become upregulated if you inhibit it. So it's needed to maintain integrity of repression complexes, which starts to make you think about how intimately related BCL6 might be to stress pathways. Looking further at the BCL6 locus, it turns out that there are 3 heat shock elements in the BCL6 promoter that are very highly conserved throughout evolution. These heat shock elements are binding sites for the master regulator of stress response, HSF1, heat shock factor 1. And HSF1, in fact, binds to these sites. And I'll tell you about some work by Thairo Fernando, who was a student in the laboratory. And she showed, I'm going to give you a few examples of data, just to illustrate some of the key points. So she showed, for example, if in HSF1 heterozygous mice, if you heat shock them, that there is failure to, that BCL6 gets induced normally, first of all. And induction is impaired throughout the mouse in different tissues if they have haploinsufficiency of HSF1. And here, HSF1 was knocked down in fibroblasts, and fibroblasts also will respond by inducing BCL6 if you heat shock them, but knocking down HSF1 prevents this. So it looks like HSF1, BCL6 form an axis. And of course, BCL6 didn't evolve. Evolution didn't know that we had to have germinal centers. And BCL6 came around before those that evolved. Yes, so it evolved for some other purpose initially, and eventually became used for that. And the question is, why did it evolve? And I think this stress response aspect may be one of the reasons why this is. And since I was little, I loved going to the museums and looking at dinosaurs and evolutionary things and thinking about, Darwin was my hero when I was 5 years old. And so I was naturally drawn to trying to ask these kinds of questions for BCL6 and wonder, why did vertebrates actually get this factor? And indeed, we tracked back across evolution and find that BCL6 appeared during the Cambrian explosion about 500 million years ago, at the point where the first jawless vertebrates appeared in evolution. These are called ignathosomes. And they have given rise to the present-day lampreys. And once it evolved in these initial early vertebrates, it became then inherited by all subsequent vertebrates after that. Lampreys, by the way, are very cool animals. This is how they look. They have these wonderful suckers. They infest the Great Lakes in the United States. They actually are a hazard because they're destroying fish. Once they get in, they have to be culled. So they're pretty vicious things. Even though they're very ancient, they're still very good at what they do. And there's a immunologist at Emory University named Max Cooper who has been studying immunology in lampreys. And you'd say, okay, that is way arcane, stop it. But it's actually interesting because lampreys they have a humoral immune response without having B cells or T cells. At that point, you're like, what? Yes, but they actually have a humoral immune system without having B cells and T cells, but they have other cells that carry out similar functions to that, which Max and others who study this have called the name VLRA, VLRC, VLRB cells. And the VLRB cells, they're not B cells, but they do, these are the ones that they express a leucine repeat protein that, so lampreys don't have antibody, they have no immunoglobulin like all of the vertebrates do. They have another gene that's called LRP that has theoretical combinatorial complexity at the same order of immunoglobulin, and which they think is the immunoglobulin equivalent for the jawless vertebrates. And VLRBs are the cells that express and supposedly mutate them, and Max had this paper in Nature which showed that BCL6 is very highly upregulated also in these VLRB cells that are not B cells, but they also have a lot of BCL6. So that led us to wonder, okay, did BCL6, with all the stress-related things, did BCL6 perhaps evolve as a critical downstream protein in stress response pathways? Is that possible? And so to ask that question, the first thing we did was to obtain cells from all these different vertebrates across evolution. This over here is lamprey and fish, birds, obviously. And then we just heat shocked them, looked at mRNA for BCL6, and showed that in all cases when you stress cells, BCL6 goes up. I won't show the Western blots, but actually the Western blots work because the B2B domain is actually perfectly conserved at the epitope that one of the antibodies is for, so you can show by Western blots too. So, okay, so how exactly does it fit into stress response? Obviously, if it's a transcription factor downstream of HSF1, it won't be an immediate effect. You're talking about, two independent ways of transcription that are not going to protect cells up front from stress. And so to ask that question, we performed a series of experiments with sequential stress. So we took cells and measured survival after acute stress or survival after repeated stress. And as you're probably aware, HSF1 famously is the factor that mediates acquisition of stress tolerance, which means that cells, when confronted with a severe stress, you may kill a lot of them, but when they recover and you go back with that same stress, they won't die. That's because HSF1 adapts them to that stress. And without HSF1, that doesn't happen. And so the question is, where does BCL6 fit into this series of events? And here to show you the first set of experiments. And we're looking here at BCL6 wild type and knockout cells. And here we're looking in this light color at the initial stress and in the red color at the adaptation. And so if you look at the initial stress, whether you're knockout or wild type, you have the same amount of cells being killed by stress. Now the wild type cells, now the second time around, are not killed as much, but the mutant cells are killed equally well the second time as the first time, which tells that BCL6 is needed for the adaptive stress response of HSF1 in vertebrate cells. You can see the same thing in mice. This is knockout mice, but in zebrafish, with the help of Todd Evans, we did morpholinos for BCL6 and showed the same thing. Now, looking a little bit closer at BCL6 evolutionary conservation by Shannon entropy, we observe that the hotspots for conservation are inside the B2B domain at certain sites, in the zinc fingers, and also a little bit over here in the middle repression domain. And if you map by Shannon entropy the surface of the BCL6 B2B domain, with blue being the highest conserved, it becomes readily apparent that there's extremely high conservation, 100% conservation, of all the residues that make contact with the SMART and N-core and B-core peptides. So that part of BCL6 is the part that has not varied from the time that we were little worm-like jawless fishes. It's maintained the same. And SMART N-core predate BCL6, but the peptide they bind to BCL6 with has remained invariant from the time of the Cambrian explosion. And this peptide has evolved with or co-evolved with BCL6. Now, can you show that suggests that this complex here may be the module that actually mediates that stress adaptation? Yep, that's maybe exactly the biochemical explanation for this. To test that, we're fortunate, since we can't knock down BCL6 in lampreys and lizards and all those things, but we do have the peptides that bind to this conserved site. You can actually do the functional experiment in all these strange animals, which I loved and my student hated. So here we took that, remember I talked to you about that B2B mutant mouse at the beginning that had that phenotype with loss of germinal centers. So if we take that same mouse again and do the stress test to it, what we observe is that, I told you before that BCL6 knockout mice don't adapt. The B2B mutant mice don't adapt either. So it shows you that that point mutation abrogates stress adaptation. Now over here we use the peptides, and same thing, that with the peptides present, you rescue the cells from being able to tolerate the second exposure. The same thing happened in chickens, the same thing happened in lizards, and the same thing happened in lamprey cells, which we got from Max Cooper's senior postdoc, and now the person who's replaced him as PI at that laboratory. But if you do it in flies that have no BCL6, you don't see that effect at all. So it's a vertebrate-specific effect that's mediated through the B2B domain of BCL6 because of that conservation of sequence there. And so now thinking about it from the evolutionary standpoint, vertebrates are animals that, many of which they have a lot of disadvantages compared to other organisms, that they reproduce often slower, they often will have less progeny, they've evolved to have longer lifespans, many of them, and so probably are require mechanisms, or novel mechanisms, to protect and adapt to stress. That's probably more fundamental to the species surviving than perhaps other organisms. And so, that stress adaptation pathway has been kind of taken advantage of by the immune system for the evolution of cells that acquire massive stress, and therefore have really co-opted these stress mechanisms to protect themselves from the severe effects of stress. And in the germinal center reaction, we talked about all the checkpoints that have to go off. All that is obviously preventing cells and maintaining their survival under those circumstances. And probably what's happened in the jawed and jawless fishes is that BCL6 has enabled convergent evolution of a humoral immune response based on immunoglobulins in jawed vertebrates and leucine repeat proteins in jawless vertebrates, which have both been able to generate independently an immune system that involves high stress conditions thanks to the presence of BCL6, which didn't evolve for that purpose, but which was hijacked in both cases as these species advanced and developed further. Now, so getting back to that first slide, yes, BCL6 is kind of the weapon that's enabled this possible for us to actually do that. So this paradoxical thing of being able to have a cell type with accelerated evolution in a vertebrate is made possible by this protein that evolved to enable stress tolerance and give us very stress tolerant cells in this case that can do that kind of thing. Now, the last point is that you would think that that's somewhat of a dangerous weapon, because if it can make cells super tolerant to stress, then, for example, if that finding was discovered by solid tumors, for example, that would make them pretty bad actors. But of course, since all cell types can induce BCL6 under stress, then you would think that maybe solid tumors would have figured that out. And in fact, BCL6 turns out to be very widely expressed across most tumors. It's very highly expressed. Of course, the top one is DLBCL, but also gliomas, ovarian, lung especially, breast cancer. In fact, many tumors have amplifications of the BCL6 locus that cause it to be overexpressed genetically. And if you look across solid tumors at outcomes, looking at TCGA data, BCL6 high expression is in almost all solid tumors linked to inferior outcome, which again makes sense for cells that would become more resilient. I'd like to show you just two examples of data here. There's a linear correlation in solid tumor cells between BCL6 and heat shock proteins that are downstream of HSF1, showing that it's part of that same program. And if you knock down HSF1 is widely expressed, of course, in solid tumor cells. You knock it down, and BCL6 goes down as well in solid tumor cells. And administering BCL6 inhibitors to solid tumor cells makes them more sensitive to chemotherapy, because, of course, there's effects on turning down these stress pathways. We actually have now found one of the key genes that's downstream for stress response, which happens to be another interesting gene called TOX. That's also mutated in tumors. And that's the kind of third step. So you have HSF1 to BCL6 to TOX, involved in protecting the genome and enabling cells to survive stress. So it seems like solid tumors indeed have learned the trick of hijacking BCL6 as part of an evolutionary conserved stress response that all cells have. And so one of the stresses the tumor cells have to survive is exposure to chemotherapy drugs or other drugs or targeted drugs that we give them. It doesn't matter if they're chemotherapy or not. And so the possibility of using BCL6 targeting drugs together with this may be a way to actually remove or take away some of the plasticity of cells to tolerate these kinds of stresses, and perhaps will be applicable beyond the area of lymphoma. So I think I've stayed on time. The work that I showed that was unpublished, all the evolution part was Thairo Fernando, and all the HDAC3 part was Patricia Mondello, and this is our lab. The stairs at Cornell. I want to also acknowledge our many collaborators in these different projects and funding mechanisms. Thank you very much.